Today, I will be talking about SARS-CoV-2 variants. COVID-19 is caused by a virus named Severe Acute Respiratory Syndrome Coronavirus 2, also called SARS-CoV-2. Structurally, the virus is made of ribonucleic acid or RNA, which is enclosed by a nucleocapsid or N protein. The viral nucleocapsid lies within the cytoplasm of the virus, surrounded by a bilipid layer or envelope layer. The envelope layer consists of three structural proteins, including the spike or S protein, membrane or M protein, and envelope or E protein. The viral genome is positive sense, with a 5-cap structure, and a 3-polyadenylate tail structure. The genome consists of about 30,000 chains of nucleotide sequences, which include a phosphate group, a 5-carbon sugar, and a nitrogenous base of adenine, guanine, cytosine and uracil, represented by the alphabets A, G, C, and U. The sugar-phosphate groups serve as a backbone for the nitrogenous bases. Viral replication occurs within cells following binding of the viral spike protein with ACE2 receptors found in various organs, such as the lungs, heart, kidneys, and gastrointestinal tract. Within the host, the viral life cycle begins with attachment to ACE2 receptor, entry into host cell, uncoding of viral RNA, production of viral proteins, replication of virus, assembly of virus via the endoplasmic reticulum and Golgi apparatus, fusion of assembled virus with host cell membrane, and then release of new virus. The genome replication process of viral infections involves creating complementary copies of virus through a process mediated by an RNA polymerase enzyme. The RNA polymerase moves along the genome creating complementary nucleotide sequences. This way, the virus can make more copies of itself. However, the iterative process of replication is error-prone and could lead to changes in the original nucleotide sequence. The change in nucleotide sequence during viral replication is called mutation. Mutations may arise from substitutions of one or more base pairs, insertion of new base pairs, and deletion of existing base pairs. Since all these mutations results in viral particles that are derived from one virus but have some differences in their genetic makeup, they are called covariants. Although not reported in SARS-CoV-2, some viral variants could recombine to form an entirely new strain. Consequently, SARS-CoV-2 variants emerge from error-prone iterative replication processes that could lead to emergence of thousands of covariants of different lineages. A lineage consists of a group of variants with similar characteristics. The World Health Organization has classified SARS-CoV-2 variants into variants under monitoring, variants of interest and variants of concern. A variant under monitoring is a SARS-CoV-2 variant with genetic changes that are suspected to affect virus characteristics but require enhanced monitoring and repeat assessment pending new evidence. Variants of concern has been associated with one or more of the following changes: increase in transmissibility or detrimental change in COVID-19 epidemiology, increase in virulence or change in clinical disease presentation, decrease in effectiveness of public health and social measures or available diagnostics, vaccines, and therapeutics. Variants of interest are COVID-19 variants with genetic changes that are predicted or known to affect virus characteristics described under variants under concern and identified to cause significant community transmission or multiple COVID-19 clusters. To resolve the complexity and stigma associated with the naming of SARS-CoV-2 variants, the World Health Organization has adopted the use of Greek alphabets as labels for emerging variants. As of October 2021, the following were classified as variants of concern by the WHO: Alpha, Beta, Gamma and Delta variants. The Delta variant, B1617, is responsible for the ongoing transmission of COVID-19 globally. Examples of variants classified under variants of interest include Lambda and Mu. Variants under monitoring may be readily added or removed, and as such they are not assigned WHO labels at this time. The variants listed under each may be reclassified in future depending on changes to their risk to public health. The COVID-19 vaccines approved for use today are known to be protective against severe COVID-19, hospitalization and death. These COVID-19 vaccines elicit broad immune responses involving a range of antibodies and cells. As such, they provide some protection against all known variants of COVID-19. However, the emergence of new variants or strains that may be completely unresponsive to existing vaccines is a source of concern. In such cases, there might be need to develop completely new vaccines that could guarantee protection against these new strains. Variants arise from repeated viral mutations. Stopping or reducing viral replication and person-to-person transmission remains key to halting the emergence of new SARS CoV2 variants. Current measures to reduce transmission, including frequent hand washing, wearing a mask, physical distancing, good ventilation and avoiding crowded places or closed settings, will reduce the rate of transmission and spread of the virus, and consequently mitigate the opportunities of the virus to mutate. However, vaccination remains the most effective strategy to halt the current COVID-19 pandemic, and to prevent emergence of new viral variants. High COVID-19 vaccination coverage will protect the population against COVID-19, interrupt transmission of the virus, reduce the opportunities for viral mutation, and ultimately prevent emergence of new SARS CoV2 variants. Protect yourself. Protect your family and community. Get your COVID-19 vaccine today.